I am CA Shirish Vyas. I am teaching.
Hello students, am I audible and visible? Are we live? Okay. Am I audible? Is there any disturbance? No, right? Good evening. Good afternoon. Yeah, we are between, we are going to be, by the class ending time, will become evening. We'll start in the mid of afternoon, right? Okay. Students, am I clearly audible? Let me know quickly in the chat box. Am I clearly audible? There's no disturbance, right? Okay. Okay. So ask your friends to join quickly. Avengers batch students, share the link quickly and ask them to join. Okay? Yes. Okay. Yes. Okay, fine. So, hello students, welcome to Unacademy NEET English channel. Myself Gopika, your biology educator. Okay? So, we were doing photosynthesis in higher plants, right? Yes. So, good evening to all of you. I am so happy that, before I say anything, I want all of you to share the link quickly to your friends and ask them to join, okay? Because you know these concepts are difficult, you know these concepts are not easy to understand, right? Yes. Okay. So, we'll start the session. Are you all ready? We'll start the session, right? So, students, I don't know how many of you got to know about this, okay? So, look at this. Yesterday, many of you were asking me, ma'am, what about the mock series, right? What about the Indian test series, right? Yes, I have come up with all the information because yesterday I couldn't give you all a lot of information. So, today I thought I will give you a detail, okay? Because I think many of you are trying to write mock test because most of you are studying through YouTube and want to attend series, right? Mock test series. So, I thought I will come up with a detailed video to let you know what is happening, right students? So, I have taken whatever questions you have asked yesterday and I have come up with the video. I will give you more clarity on this. So, students, we are going to start our Unacademy All Indian Test Series, okay? And you can look here, UAIT's NEET UG 24 pen paper based for class 12 and droppers is for 5999, okay? It's for 5999. 1 minute, I'll use the pen. Yes, and here for again for your 12th and droppers for NEET UG, this is online, this is 3999, okay, 3999. And here we have the questions that was asked most yesterday for class 11 students, right? We have it for 1999, okay students? So, your all Indian test series, we are starting it again, right? We are starting it and these are the prices. So, if any of you want to join, even if you are a student who is only focusing on YouTube and trying to write your exams, but students writing tests, right? All Indian mock series are very, very important because you need to know where you stand, right? Just by studying or every day attending live classes is not the solution for cracking your NEET when there are lakhs of students writing, okay? So, your focus should be to see where you stand, okay, where you stand. So, for students who wasn't aware of this, I have come up with the video. So, please quickly discuss with your parents and join because seats are limited, right? For this price, seats will be going very fast. And we have also started our unsat, okay? We are starting to take unsat reopening registrations, okay? And I don't have to tell you anything about unsat because you already know that we will be coming with detailed where did you go wrong, what went wrong, where do you stand, all these detailed lectures will be given by the teachers. So, if you are a part of this, you will exactly know where do you stand, right? Your JEE, even if you are an JEE student, even if you are a NEET student, very important students to participate or to take part in such things to know where you stand, okay? And see, look at the timings, so you do not miss. Right now only make a note, right? Link is in the description box. All the link is in the description box. Okay, just go to the description box and you can click on the link. Okay, students, and if you want to join Avengers 3.0 for just 3,999, the link is again in the description box, because we are relaunching Avengers 3.0. Okay, and for any kind of discount, use my code. Okay, use my code. And what's my code? GOPIKA, right? GOPIKA, and you're going to get maximum discounts. Students, look at the dates. Look at the dates, because last time we had very good majority of students participating. So look at the date. It's from October 16th to 21st. 8 p.m. onwards. Okay, 8 p.m. onwards. Those who are new to this channel and who doesn't know what UNSAT is, students, we will be conducting proper mock test, right? That will be starting from 16th to 21st. And do not worry, because once you finish the exam, we teachers will come up with a proper video to explain you what went wrong. Okay, what went wrong. Got it? So students, your Avengers 3.0 will start quickly, and the seats are very limited. Okay, and the prices also are very less and amazing. So those who are trying to study for NEET, students, 6 months is there, and we will assure you that you can crack NEET. Okay, do not forget to join. Fine, the link is in the description box. And we have so many things. I already mentioned you people. We are getting educator practice revisions. We are getting PYQs. We are also getting last 15 years PYQs, chapter wise PYQs, and also mind maps and super 30 quizzes. Where are you going to get everything together? Here, right? Here. Okay. So do not forget, you're getting all of this. And you're getting NEET Key Brahmastra seal. You're getting everything again, right? You're getting your mind maps and flashcards. I have seen a lot of you students asking for mind maps and flashcards, especially for chemistry and everything. You will get everything here. Okay, look here. All this is available here, right here. What you have to do? You only have to enroll. Okay. And Telegram channel also, Unacademy NEET English Telegram channel. Just go to Telegram, type Unacademy NEET English. 10,000 plus students are there. Okay, do not get confused and join the wrong groups. Okay, so students, clear? We are relaunching UNSAT, and this is the right time for you to join. Okay, right time for you to join, because I saw a lot of students asking, ma'am, YouTube video. I'm watching all the YouTube videos. Students, watching just YouTube videos is not the right way. Okay, right way. Okay, so quickly enroll now. Use my code GOPIKA, GOPIKA, and get your discounts. Okay, why I'm telling so many times? Because the seats are very limited. Okay, being said that, I hope this is clear, because yesterday I got a lot of questions and I was not able to clarify everything. So today I thought I will spend some time and give a clarity on those who were asking about it. So those who were asking, I hope everything is clear, and whatever links you want, everything is in the description box. And to join Avengers 3.0, it is in the comment. Okay, it's pinned in the comment. All of you can see, I hope. Yes. So let's start photosynthesis in higher plants. Okay, are you all ready? Is every doubts clear about your mock series? May I know in the comments chat box if all the doubts are cleared, whatever you asked yesterday? Yes or no? Yes. Quickly let me know. Yes. Okay. Students, let me know so that I can proceed. Okay. Whatever doubts you are asking, I am bringing it in the next video, right? So yesterday whatever you asked, we are, yes, okay. So students, new syllabus, everything is also put up by Ambika ma'am and myself. We both have come up with a video and we have clarified all your doubts about it. Okay, all your doubts about it. So the only thing now you have to go is click the description box, see all the videos. Okay, fine. Let's start photosynthesis in higher plants. Students, yesterday we did chloroplast, we saw all the early experiments, and we also reached up to the graph, right? We reached up to the graph where we are explaining about the pigments, right? At what rate or at what wavelength will the pigments absorb, right? Will the pigments absorb? So here I have put the structure of chloroplast because it's very important, and I want all of you to keep seeing it again and again, right? So we have outer membrane, inner membrane, stroma, lamella, grana, again stroma. This is the stroma, the matrix. Okay, ribosome, that is 70S ribosome. We have starch granules and lipid droplets. Okay, students, clear? whoever was there in yesterday's class and understood everything what we did up to yesterday, quickly like the session. Quickly like the session and let me know. Then only I'm going to proceed, okay? Otherwise, I won't proceed. Yes, okay. Students, share the link also. Share the link quickly with your friends, okay? So that more students can join. Yes. Okay, shall we start? Students, please, from now on, just from this minute, I want all of you to take your notebooks, pen, pencil and be ready, because we are going to break down a big concept, right? We are going to break down a very big concept and I'm going to try to make it very easy for you all, okay? That is your photophosphorylation, right? Cyclic and non-cyclic, okay? Yes. So, here we did yesterday up to the graph, right? We did the graph where we understood that the blue area and the red region, right? The red region will have maximum absorption, maximum action. That means at that point, the chlorophyll A, especially the chlorophyll A, is going to absorb more of the energy, right? So, yesterday I wanted to go through this, so today we will just go through it, students, quickly. This is your NCERT lines after the graph which is given, okay? So the terms seems a little complicated, so I thought I will make it, I'll break it down for you. So these graphs together show that the most of the photosynthesis takes place in the blue and in the red region of the spectrum. Some photosynthesis does take place at other wavelengths of the visible spectrum. So visible spectrum is from where? 400 to 700 nanometer, right? So between that also some places it's happening, okay? Because we do not forget, we should not forget about who, we should not forget about your accessory pigments, right? We should, now we are only focusing on our chlorophyll A, but there is also our accessory pigments, okay? So though the chlorophyll is a major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophyll, carotenoids are also important. Yesterday I told you, students, yesterday I told you, right? I told you very majorly I told you that you should not forget about your accessory pigments, right? My voice is clear, no? Is it only Surya who is finding it problem? I don't think there is a problem with my voice. It's clear, right, students? Yes, okay. Yes, okay. So students, which are also called as accessory pigments, very, very important, accessory pigments. Now I am telling you, you know why I am telling you this again? Because when you are going to study for, your photophosphorylation, all these terms will come again. That time you shouldn't be confused, okay? And absorb light and transfer the energy to chlorophyll A. So what do? They absorb the light, the accessory pigments. What do accessory pigments do? They do something like, they, like, students, it's like a hero and the hero's friends, or you know, the co-actors, right? The co-actors. Now one hero will be there, he'll have other three brothers, right? So what is he going to do? These three brothers are going to help this hero to be a, you know, a better hero, right? I don't know how to say it, but yeah, to a better person. Same way, chlorophyll A is the major pigment or the master pigment. The rest of them are accessory pigments. What are they going to do? They are going to trap the energy and give it to chlorophyll A. Okay, yes, and not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect the chlorophyll A from photooxidation. I told you this yesterday, photooxidation. Okay, yes. So what do? They absorb the light. This accessory pigments will absorb the light and give it, or absorb the energy and give it to chlorophyll A. Okay, I'll tell you in detail about that. Do not worry. Okay, detail about that. Do not worry, students. Very, very important. I want all of you to write whatever I am writing, okay? Because I won't give you notes for this until you write. First you should tell me, ma'am, I wrote, then I'll give you notes. Students, photosynthesis. Yesterday we discussed photosynthesis has two processes, right? Two processes. What are the processes? They have a light dependent process and a light independent process, right? Light dependent and light independent, right? So I can also call them, what can I call light dependent as? I can also call it as light reaction, right? I can also call it as light reaction. I can call light independent as dark reaction. Okay, dark reaction. Now, I can also call light dependent. I can also call light dependent reaction as, okay, I want to also call it as photophosphorylation. Such a long name, no? Photophosphorylation. Students, do not fall unconscious just reading the name. I will break it down for you. Okay, so I'm going to write that down. So this much is clear. Photosynthesis has two processes: light dependent, which is also called as light reaction; light independent, this is also called as dark reaction. I told you very clearly yesterday, do not think that dark reaction means it is taking place in the dark. That means it does not need light directly. Okay, indirectly it requires light. Now let's take photophosphorylation. Okay, photophosphorylation. Students, photophosphorylation, what do you understand? Photo, okay, that means what? Light. Phosphorylation means adding phosphate, right? Adding phosphate. Now I want to, so what is happening? Not me. The photophosphorylation can be of two types, okay? Non-cyclic and cyclic. So much is clear. So much is clear, right? So photophosphorylation is divided into non-cyclic and cyclic. Okay. Now, light photophosphorylation, light and adding phosphate. Now, what are we going to do? Right? What are we going to do? We are going to study first non-cyclic, because this is more dominant. Why I'll tell you it is more dominant? It is because during this reaction we are synthesizing ATP as well as NADPH. Okay, this is why this is a more predominant one. Okay, right? Now here cyclic, we are only synthesizing ATP. Now what do we, why do we have to study this? Understand students, dark reaction is dependent on, that means for dark reaction to take place we need ATP and NADPH, and both of this is provided by the non-cyclic, ATP is provided by the cyclic. Okay, fine, this much is clear. All of you made a note? All of you made a note? Can I proceed? Yes or no? Yes or no? Yaar, where is the Josh, yaar? All of you are sleeping, huh? All of you are sleeping, huh? Students, don't worry, I will tell you the locations. Okay, now let's not get into the locations. Let's go slow. Let's go slow. The process is to understand. The process is not to finish the chapter. Okay, yes, good. Okay, now this is done. Now we are going to study non-cyclic. Okay, now we are going to study non-cyclic. Very, very important. Very important, and hardly some students understand this when the teacher is, usually a lecture is happening in class, right? Okay, so non-cyclic. Students, let's break it down. Let's think it is very easy. Now what is going to happen? We know that two types of photosystems are there, right? Two types of photosystems are there. What are they? PS1 and PS2. Okay, these photosystems are not, the naming is not based on the, you know, what they are doing. It is mainly based on their inventions. Okay, it is not based on their functions, right? So PS1 and PS2. Now photosystem 1 and photosystem 2. Now what is photosystem? They are basically a biochemical reaction where it helps for photosynthesis to happen, for electrons to move, for all of that, right? Yes, yes. What is happening in the chat box? Students, no spam. Focus here, it's a very, very important concept. Okay, so this much is clear. So in non-cyclic reaction, we have, so I'll start from here. We have PS. What I'm trying to draw? I'm trying to draw your chlorophyll A with the accessory pigments. Okay, chlorophyll A with your accessory pigments. So you have chlorophyll A and you have carotenoids, xanthophylls, everything. So what is going to happen? As soon as light, right? As soon as light is emitted on them, they are going to absorb the energy. So these people also will absorb the energy and give it to chlorophyll A. Just now we studied that. Agreed, all of you? Yes. Now, which is the system that is coming in place? Here we have PS2 that have 680 nanometer. That means chlorophyll A will absorb energy at 680 nanometer, right? 680 nanometer. Now, what's going to happen? Now, so much energy, right? So much energy is being emitted. So much energy is being absorbed by your chlorophyll A, right? Now, what is going to happen? This chlorophyll A will release a pair of electron, right? It is going to release a pair of electron, right? It is going to release a pair of electron because of the exciting energy. Now, that electron will be accepted by something called as PEA. Okay. Now, I'm just going to draw a few boxes. I want all of you to draw with me. Okay, but because by the end of this, I want all of you to be clear with this. Okay, PEA. Now, PEA stands for primary electron acceptor. Okay. Now, from this electron has been released, it is going from primary electron acceptor to PQ. Okay, CY and PC. Now, I'll tell you what is all this. Students, basically this is an electron transport chain. What is happening? The electron, here the chlorophyll A absorbed the sunlight. This carotenoid xanthophylls, the accessory pigments also absorb the sunlight and they gave it to chlorophyll A. Now, chlorophyll A, what happened? The energy got excited, right? The energy got excited and the electron, a pair of electron was tried to escape. Now, what happens? Now, you people are just sitting and watching live, right? You haven't joined any batches, you haven't attempted any mock series. So, here for you, no excitation is there. That means no energy is there, right? Once you join batch, once you join mock series, what is going to happen? Excitation is going to be more. That time, what is going to happen? You're going to come up with good results. And this is your MBBS. This is the place where you have to be, right? This is all your teachers trying to help you, but you people are only attending live and not joining any batches, doing anything. So, here energy is low. So, will you reach here? No. Will you reach here? No. Okay. So, you will not reach MBBS. So, fine. Okay. Understood. This is the easiest example. Okay. Now, students, this is an electrode transport chain. So, I will write what is all this. Okay. I'll write it in this corner. PQ stands for wait, 1 second. Okay. So, PQ plastoquinone. Okay. And what have we written next? CY. CY stands for cytochrome. Okay. Cytochrome complex. Students, very important. If you're not making a note of this, it's really a loss. Okay, it's really a loss. Okay, and PC is plastocyanin. Yes. So, students who have really prepared are very consistent. What is going to happen? They are going to reach here. From there, they are going to move to one phases of their life, right? So, here this is an electron transport chain. Okay. PQ stands for plastoquinone, CY stands for cytochrome complex and PC stands for plastocyanin. Clear? So, what is going to happen? Our electron is going to come from here, move here, okay, and also move here. That means, students, the energy is so much that the electron is continuously moving, okay, continuously moving, but very important, it is not going to come back, okay, it's not going to come back to your reaction center. This is called the reaction center. It is not going to come back to your PS2. Okay, it's not going to come back to PS2. It is continuously moving. Now, what happens when it is transferred from PQ? Okay, when it is transferred from PQ to CY, that is your plastoquinone to cytochrome. 1 second. Yeah. When it is moved from your PQ to CY, there is some extra energy. That extra energy will be utilized to convert ADP to ATP. It will be used to convert ADP to ATP. How is it happening? This is where your phosphorylation is happening. All of you understood how did we get the name photophosphorylation? Yes or no? Yes or no? Hi Vashini, you have joined the right place. You have come to the right place. Okay. I'm so happy that there are a lot of new students. Okay, new students. So students, this channel is definitely the right place for you to prepare for NEET. Okay, so just one thing, just be focused. Okay, just be focused, don't get distracted. Okay, so this much is clear. Students, very important, very important concept. Okay, so what is happening here? There was some extra energy in the CY that is utilized to convert ATP to ADP to ATP. Okay, by adding a phosphate group. Clear? Yes. Okay. So here we have a phosphorylation with the help of light. Okay, with the help of light. Now what is happening? Now here PC, the electron has reached PC. Now the electron has to move because the energy is more. Now what is going to happen? This electron is going to move again. Okay, going to move again. I have to reduce that, no? 1 minute. Okay, the energy is going to again move to your PS. Okay, we are going to see our PS1, that is 700 nanometer. Okay, students, PS1 700 nanometer. Yes, what do you want me to repeat? Okay, so what is happening, students? Here the energy is more. It is not going to stop. It is going to the PS1. It is going to PS1 from PS2, right? So it is not going back to PS2, but is going to PS1. Now here who is there? Chlorophyll A is there. Carotenoids are there, right? Okay. Now the electron has gone here. From here it has to go elsewhere. Okay. So I'll just erase this. I hope all of you made a note. I'll just write it smaller here, so we can finish the system here. Otherwise you will not understand. Okay, I don't want to break the cycle. So I'll just write. Okay, now what is happening, students? Did the electron stop? Did the electron stop? No. Here also what is happening? Light is there. There is light energy here also, right? Everything is happening in the presence of sunlight. Now here also these accessory pigments are going to absorb the light and give it to the chlorophyll A, and the chlorophyll A is going to get very excited and again release electrons. Okay, release electrons, and at the same place we are going to have your PEA, that is your primary electron acceptor. Primary electron acceptor. We will revise it. Okay, don't worry, but so much is clear. Primary electron acceptor will be there, right? From primary electron acceptor, students, did you make a note of this? Can I erase it? I'll erase this. Okay. Yes. Students, from here it is going to a new term that you are going to study is FD. Okay, FD. Okay, FD stands for ferredoxin. Stands for ferredoxin. Okay, ferredoxin. From ferredoxin, it is going to form your. We are going to see our NADP. Okay, from ferredoxin, it is going to move to NADP. Do not worry, I will repeat it. We will revise the whole thing. Let me first complete it. Okay, NADP. Clear? Students, do not worry. I know the concept is complicated. I will revise it for you. Okay, NADP. Students, from the ferredoxin, what is moving? Our electrons are moving. Okay, electron is still moving. See, here also it is moving. Here also it is going to move. Now, this NADP. What are we going to convert NADP as? NADPH. NADPH. But for this we need a proton. So here we will have NADP reductase. Okay, NADP reductase. Okay. Now all of you tell me one thing. Now we lost electrons from here. Don't we have to regain that electron? Don't we have to regain electron? When we lost something, do we have to regain or we don't have to regain? Tell me. Do we have to regain or not regain? Quickly, I am waiting for your reply. Students, yes, regain, right? So, to regain, what we lost, the electron that we lost here, no? What is going to happen? Photolysis is going to happen. Students, what is going to happen? Photolysis, okay? Photolysis is going to happen. I'll just write it here. PS2 680 nanometer. Okay. Now, photolysis is going to happen. Photolysis of what? Photolysis of what? Water, right? So, I am going to break the water molecule. So, I get the electron that I lost. Okay. Now, look here. I have broken down. That means photolysis of water has taken place and we got a proton, electron and a nascent oxygen. Now, what is going to happen? This electron is going to come here. All of you understood? All of you understood? This electron, photolysis, the water that is broken down is going to come to the PS2 so that the lost electron can be regained. How many of you understood this? Yes. Okay, great. Okay, now students, we have a proton. Now, one person is waiting there to convert it to NADPH. So, can I use this proton to convert NADP to NADPH? Tell me. Yes, right? So, I am going to use this water molecule to convert my NADP to NADPH. Okay? Convert NADPH2 because I am going to add this. Look here, I am going to draw a box around it so you understand. I am going to take the proton, right, and convert my NADP to NADPH2. Now, this, what did I get? What did I get? Students, whatever I have drawn in green, right, whatever I have drawn in green is the output that we have got from a non-cyclic phosphorylation. Non-cyclic phosphorylation. Okay, can we quickly revise? Can we quickly revise? Are you all ready? Put my favorite emoji and tell if you understood so much but you want a quick revision. Tell me quick, very quick. Yes. I want more response here. If there is no Josh when I am teaching such a complicated concept, then I am not going to repeat. Okay. Now, it's like this, right? I don't want you people to feel like this is an online class. That's why I am asking you people to respond. So, quickly respond. More response I want, more response. Then only I am going to, no, NADP, see, this H plus proton goes there to NADP, converts it to NADPH2 with the help of NADP reductase. Only if NADP reductase is there, this whole thing is going to happen. Do not get it confused because NADP reductase is the enzyme. Enzyme does not take part in the reaction, it will only speed up the reaction, right? Okay, got it, Anusha? Got it? Do not get it confused. That is why photolysis is very needed. So, let us do a quick revision. Are you all ready? I want all of you to answer very quickly so that we can finish it fast, right? Yes. So, let us start non-cyclic photophosphorylation. Now, what is going to happen? We have a reaction center. That means we have a chlorophyll A with all the accessory pigments. Okay. I will write it here. Okay, reaction center. Okay, so this is the reaction center where we have chlorophyll A, right? It starts with PS2, that is 680 nanometer, PS2 system, right? Photosystem. Now, what has happened? They absorbed all the sunlight. And what happened? This chlorophyll A got excited, right? We tell no excitation state. Now, they released a pair, or the chlorophyll A released a pair of electron that was accepted by the primary electron acceptor, which is known as PEA. Okay. Then we are going through a series of electron transport cycle, right? Electron transport cycle. So, from the primary electron acceptor, the electron is moving to PQ, that is your plastoquinone. Okay. Is it okay if I write it here? plastoquinone. Okay. From the plastoquinone, it is the electron is again moving to our CY. What is our CY? Cytochrome complex. Okay. Cytochrome complex. Right. From the cytochrome complex, okay, I'll tell you this, the lumen stroma doubt, no, I will clarify for you. Okay. Now, CY, okay, there is a cytochrome complex. From that also it is moving to our PC, right? What is our PC? Plastocyanin, right? Yes, plastocyanin. Now, students, in the CY, that is your cytochrome complex, there is some extra energy. That extra energy will be used to convert our ADP to ATP. So, in the first step, what did we synthesize? We synthesized ATP. Now, will the electron stop there? No. The electron will move to PS system 1, where we have your 700 nanometer, right? 700 nanometer. Now, from here also, they are going to absorb light, excitation is going to take place, electron is going to release. Again, our PEA, that is your primary electron acceptor, is going to accept it, pass the electron to ferredoxin. Okay, from the ferredoxin, it is going to move to our NADP, which is waiting to get, okay, your reductase. So, in the presence of NADP reductase, that is your enzyme, your NADP is waiting for what? For a proton to come, right? So, that time who is going to help? Photolysis of water is going to take place, because here we lost the electron and this is a non-cyclic photophosphorylation. That means the electron is not going to come back to the photosystem 2. That time we are lacking electrons, right? For that time what we are going to do? We are going to split the water molecules. Splitting the water molecules will lead to give rise of a proton, electron and oxygen, okay, nascent oxygen. Now, this proton will be used by NADP reductase to form NADPH2, okay, NADPH2, and this electron, right, will go back to our PS system 2, okay, and this oxygen we got, right, this oxygen we got. Now, tell me, throughout the process, what and all did we synthesize? I want all of you to answer quickly. What and all did we synthesize? During this whole non-cyclic photophosphorylation, what and all did we synthesize? Tell me quickly. Yes, ATP, NADPH2, yes, very good, and oxygen. Great. Understood? Is this clear to all of you? Is this clear to all of you? Because most of them feels this concept very, very difficult. Is it clear, students? Yes or no? Yes? Easy? Easy? Is it easy or not? I want to know that also. Understood? Easy? Yes. Okay. Now, site of reaction, where and all these reactions are happening. So, our non-cyclic reaction, I'll put NC, students, is happening in the thylakoids. It's happening in the thylakoids, okay. Non-cyclic reaction is happening in the thylakoids. Then what do we have? Students, very, very important, why it is called non-cyclic reaction? It is because the electrons is not going back to PS system 2. Okay, that is why it's called non-cyclic. Yes, yes. Now, photolysis, okay, photolysis of water, that is breaking down or splitting of water, is taking place where? In the inner membrane, in the inner membrane of your thylakoid. Inner membrane of your thylakoid. Now, there was a doubt, right? Now, oxygen formation, that is splitting of water, is taking place in the lumen, okay, taking place in the lumen. Now, here photolysis of water is taking place in the inner membrane of thylakoid. So, here, see, if here it is happening and your non-cyclic is happening in the thylakoid, right? This, to know this, what is this non-cyclic reaction, you have to know this two site, it's very, very important, okay, this two site. That means your non-cyclic reaction is taking place in the thylakoid and your photolysis of water is taking place in the inner membrane of the thylakoid. That means if one is happening here, one is happening here. Clear? Yes. So oxygen will be produced on the lumen. So the O2 will be produced in the lumen or your nascent oxygen. Clear? Any doubts? And NADP will be in the outer membrane, because students, one more thing very important, your NADP reductase will be present in the outer membrane of the thylakoid. So do you understand, the whole thing is happening in the thylakoid, in the inner membrane, outer membrane and the thylakoid. So what is going to happen, these reactions are all going to simultaneously happen and help each other. Is it clear? So this NADP reductase is only present in the outer membrane. So this whole thing can only happen if NADP reductase is there. Clear? These locations are very clear. It's very important because for NEET exam they will ask you. So tell me that this is easy now. Non-cyclic reaction. So much I've written, no? Non-cyclic reaction, students. It looks very difficult, but isn't it easy? Isn't it easy? It is like climbing down the stairs, right? We go one stair to another stair to another stair, then we continue. So it is just like that. Yes, ma'am. Okay. So can I move on? We have to study cyclic. Way too easy. That is great. That is great. Okay. So this is what is given in your NCERT. Okay, I have explained all of this. Only thing that I didn't talk about is your LHC. That is your light harvesting complex, which is within your photosystem 1 and photosystem 2. Okay, and here antenna, one word is used. That is basically each photosystem has all the pigments and except one molecule of chlorophyll A. That means chlorophyll A will be in the center. All other accessory pigments will be together, forming a light harvesting system, also called as antenna. Okay, so your reaction center, students, you don't have to get into the depth. Okay, now I have explained all this. We will quickly understand cyclic. Okay, if you understand cyclic, then it will be easy for you to relate. Okay, now I have taken your NCERT lines. Okay, in case if you people have a doubt. Okay. Okay, students, this is not that complicated. This is very easy. Okay, yes. Rapid fire, I'll do rapid fire for all of you, but before that listen. Okay, before that listen. We will finish cyclic and then do a quick rapid fire. Okay, yes. Okay, fine. Now students, okay, yes, your non-cyclic. Okay, non-cyclic reaction is going to, non-cyclic is very simple. First of all, from the word, what do you understand? That means the electron that was released will go back to the system, right? Electron released will go back to the system or will return, right? We'll use better word, right? 1 minute. We'll shift here. Yes. Will return to the system. Cyclic means that, right? Now, very important, students, this non-cyclic has only one system. Okay, what is that one system? Look here, your PS1. What is PS1? 700 nanometer, right? 700 nanometer. So this 700 nanometer is still going to have accessory pigments. Right? Accessory pigments are going to be there. Fine. So students, this is very easy. Okay, this is compared to non-cyclic, this is simple. Okay, now what is going to happen? Okay, I'll draw it elsewhere. Yes, so here we have our chlorophyll A, we have our accessory pigments. So the moment light is there, they are going to absorb all the energy and emit it out, right? Absorb all the energy and emit it out. Yes, PS1, correct, all of you are correct. Now they are going to release the electrons. Now this electron is going to get released. Is it visible to all of you? Yes. Okay, now who is going to be waiting? Our PE, PEA is going to be waiting. Who is our PEA? Primary electron acceptor, right? From the primary electron acceptor, we are going to have FD. Who is FD, students? Yes. So, we have our FD, that is your ferredoxin. We have a PQ, which is your plastoquinin. We have a CY, that is your cytochrome complex. Again, we have plastocyanin. Okay, these terms again I am not writing because we just finished it. Now, what is happening? The electron is going, it is going here, right? It is continuously moving, right? Here also, here also, here also, and here also, right? But at two places, okay, at exactly two places, our ATP production is going to happen. Can you guess which are the two places? Which are the two spots where ATP production would have happened? One place you know from the previous. Now tell me one more. Just give a guess. Just do a guesswork, students. Where there will be extra energy. Where there will be extra energy. Wow, very good. That's amazing. Yes, Surya, very correct. FD. FD is correct, not plastocyanin, not plastocyanin. I think Pranati answered first. Okay, ferredoxin. Correct, students, very correct. So, ferredoxin, here extra energy will be there. That ADP with the help of phosphate will form ATP. Okay, and I told you this reaction will only synthesize ATP. Okay, and here also extra energy is going to be there. That energy will be used up to form ATP. So, we got two places where ATP is formed. Now, tell me, where is this electron going to go? Where this electron will go? Will it go to some other, will it go to PS2 or will it come back to PS1? Very, very important. Tell me, where will this electron go? Quick. I am writing, you people are typing, tell fast. This electron will go here. Yes, very good. Return back. Perfect. Okay, perfect. This electron will come back to our PS1. Okay, PS1. This is the reason this is called a cyclic photophosphorylation. How many of you understood? How many of you understood? Yes? Okay? Yes? All of you understood? Yes or no? Quickly let me know. Super clear. Wow. So, is it easy now? Is it easy now? Yes. All of you? Yes. So, students, quick revision. Okay, quick revision. Here we have our PS1. Okay, that means understand cyclic phosphorylation. I am going to write the most important points here. Cyclic phosphorylation only uses one system. Only uses one system, and that system is your PS1. Okay, and it does not require photolysis of water. It does not require photolysis of water because the electron is going back to the system. So, we do not do photolysis of water, right? And this PS1 is present in the stroma lamella. Stromal lamellae. This PS is present in the stromal lamella. So, where will this cyclic phosphorylation happen? Photophosphorylation happen in the PS1. And here we do not have our NADP. No NADP, right? No NADP. So, we do not care about reductase also. Is it clear? Is it clear? So, this is very, very important. Okay, very important. Okay, let's do a quick revision. So, we have our PS1. We know that cyclic photophosphorylation only happens in PS1. What is going to happen? Energy, it is going to take up the light, absorb all the energy, get excited, send a pair of electron. Electron will be accepted by your PEA, that is your primary electron acceptor. From the primary electron acceptor, it is going to come to FD, that is your ferredoxin. So, this is an electron transport chain. Okay. Now, this ferredoxin will be, that electron is moving towards your PQ, that is your plastoquinone. From plastoquinone, it is moving to your cytochrome complex. From cytochrome complex, it is moving to your plastocyanin. Okay, plastocyanin. Now, at two spots, okay, at two places, your ATP production is going to happen. Okay, ATP production is going to happen. Okay. So, that is your here at the FD, that is ADP plus PI, that is your phosphate will form ATP, and here in your cytochrome, okay, ADP plus P will give ATP, okay. So at two places, which is that cytochrome complex and FD, cytochrome complex and FD, the ATP will be formed, okay. So during this process we only synthesize, okay, only synthesize ATP. Okay, and now this electron is very excited, it is moving, right? This electron is going back to PS1, it is going back to PS1, that is why this is called a cyclic reaction. Cyclic reaction. Students, I forgot to tell one thing, in your non-cyclic photophosphorylation, no, it is called a Z scheme, okay, it is called a Z scheme. That is because why? It is starting from one place and it is ending in another place, it actually looks like an N. Right, from here again if I draw, it looks like an N, but they have told it it's a Z, right, okay. So it's a Z scheme because it's maybe because they thought that it starts from one system and ends in another system, okay. Yes, all the tech people, don't disturb the class, okay. Yes students, is cyclic and non-cyclic photophosphorylation clear? I want all of you from now on to be like, oh, that concept is so easy. Okay, that should be the goal. That is the goal. Okay, that is the goal. Yes. Quickly let me know. Today's class was only to make you understand cyclic and non-cyclic photophosphorylation, because I know how important this concept is. Yes, clear, crystal clear. Any questions if I ask, you should be able to answer. Okay, we have 5 minutes, so we will do a rapid fire. Okay, we will do a rapid fire. Clear? I am going to change the slide. All of you tell me, is it clear? Did it become easy for you, or did you already know it, whatever it is, okay? Tell me. Yes, okay, great. So I can ask you any questions? Yes, we will do a rapid fire. Are you ready for the rapid fire? Where is the Josh, yaar? You people will take my Josh also. I told you, if you are coming to my class, you have to be full energy, like energy packets you should be. You should be like this chlorophyll A and their friends. Okay, I want all of you to be the reaction center. Okay? Yes. Yes. Very good. Trisha always gets me, right? Okay. We have one person who has to be like, you know, that person is really disappointed and sad, so we will just remove that person, so his life becomes easier. Okay, yes. Okay students, fine, look here all of you. So this is clear. We will start a rapid fire quickly. Okay students, all this is your NCERT lines only. Okay, I forgot to tell you students one more thing. Those who were asking, okay, those who, I'll quickly write it down here, or else I'm going to forget. Okay students, those who were asking for pteridophytes, is it visible? No, right? One second. Pteridophytes, okay, and animal kingdom. I saw some comments, that's why I'm telling. Students, all of this is done in my special class. Special class at 10 AM I do it, and MCQ rounds, okay, MCQ of all these chapters will be done at 8 PM. Okay, this is very clear. Okay, clear? Those students who are asking in my comments, I forgot. So students, pteridophytes and gymnosperm, both pteridophytes and gymnosperm, according to your reduced syllabus, I have done it in my special class. And animal kingdom I started today, so we are going to I'm going to give you a lot of tricks to remember. Okay, you can ask my students. We finished 3 phylums with easy tricks to study the examples. You can ask my students. Students, you can put it in the comment section. We finished biomolecules also with the MCQs, and all the MCQ rounds of this set of thing will happen at 8 p.m. Okay, so if you have still not followed me on my special class, students, you are losing upon so many things. I discuss AIMS paper, I discuss NEET questions, so there is a lot of benefit out of the MCQ round, especially. Okay, PYQs, MCQs. So students, do not miss it. Yes, students, quickly, rapid fire. We have just 2 minutes. We will finish it off. First question. Ready? All of you ready? Yeah, very fast. First question. Yes, okay. Which is the first system in non-cyclic reaction? First system in your NC. Non-cyclic reaction, which is the first system? Right? Pranati, you can first enroll right now. Once you enroll, no, you will get all the information. Okay, you will get all the information on the app, because we have recorded sessions and live sessions. It depends on the teacher. So you will get all the information once you enroll. So if you still haven't enrolled, quickly enroll. The description is there. Write down. Use my code and join. After that, you will get all the updates. Clear? Do not worry. You are not going to miss out on anything. Everything will be properly mentioned, and we will also come up with a video telling about everything. Okay, students? Yes, very good. PS2. PS2. Next question. What is FD full form? What is the full form for FD? Full form for FD? Yeah, I'll teach students. I'll teach in all batches. Okay. First you people join. Then I will give you a surprise. Now you people are giving me surprise. No, now I'll give you surprise later. Okay. Ferredoxin. Yes. Yes, okay. Ferredoxin. Okay, very good. Third question. Quick. Where is NADP reductase present? Where is NADP reductase present? The enzyme is present at what site? Where is it present? Yes. Where is it present? Yes, Trisha got it right. Outer membrane. Very good. Outer membrane of the thylakoid. Okay, outer membrane. Students, fourth question. Ready? Yes? Okay. Where does photolysis of water take place? Photolysis of water takes place where? Photolysis of water takes place where? That is, oxygen will be released into the lumen, but I told you one other statement. Very good. Nageshwara has got it. Inner membrane. Very good. Last question of the rapid fire. The proton that NADP gets is from where? Okay. NADP gets some proton, no? That proton is from where? Trisha got it right. Yes, very good. H2O, splitting of water. Yes, Nandini. Very good. Splitting of water. So I hope this concept is completely clear. Cyclic and non-cyclic. Okay, students. Once this chapter gets over, do not forget we have MCQ round where I will be evaluating you, where I will be checking your progress, right? So, be always active, be regular to the class so that we can have like this amazing session. So, I hope the concept is clear. I want all of you to go back, check the video, write the notes, okay? Because this is very, very important. Once this concept is clear, the whole chapter is easy. That is why all of you were crying, ma'am, photosynthesis in higher plants is because of these concepts. Now, the concepts are clear. The only job for you is to make the notes, okay? Write down the notes, right? No, Surya, right now it's not that. Our mock test will be starting from October 16th. Okay, October 16th. So, yes, today is 18th. Yeah, you can check the app. Till 21st it will be happening, right? 21st. Yes, bye students, take care, have fun, and do not forget what I told you. Consistency is very, very important. Okay, consistency is very important. Without consistency, no one has reached anywhere, right? Okay, so quickly join the mock test series because it's very important to see where you stand in all India, right? The ranks are very important. Okay, thank you so much students, take care.